The overall goal of this procedure is to identify megabase scale DNA copy number alterations in single cells.
In order to characterize genomic heterogeneity in cell populations, it is necessary to identify genomic changes at single-cell resolution. Traditionally, this was achieved using cytologic approaches. However, these methods were limited in their sensitivity and specificity. Single-cell sequencing can detect a variety of genomic changes with enhanced sensitivity and specificity over prior approaches, but only when carefully executed. Here, we describe how to use single-cell sequencing to reliably detect megabase scale copy number alterations in single cells.
To assemble the microaspirator, begin by removing the clear plastic end from the aspirator tube assembly, and insert the narrow end into one end of a 1-foot-long PVC tubing with a 3/16 inch inner diameter. Insert the outlet of a 0.2 micrometer syringe filter into the other end of the 1-foot-long PVC tubing with a 3/16 inch inner diameter. Then insert the inlet of the 0.2 micrometer syringe filter into one end of a 6-inch long PVC tubing with a 5/16 inch inner diameter. Break a 5 milliliter plastic serological pipette at the 1 milliliter graduation and insert the broken end into the open end of the PVC tubing with the 5/16 inch inner diameter. Next, insert the outlet of the 5 milliliter plastic serological pipette into the flexible end of an aspirator tube assembly. Then use a sterile plastic tube to cover the red mouthpiece of the tube assembly for storage. Draw out a glass capillary tube to an inner diameter of 10 to 30 micrometers by positioning the middle of the tube near a flame and applying tension on either end of the tube.